Hello.
Oh,
How are you?
oh, good. I'm having breakfast.
Oh, good.
Yeah.
Hello, Lei Peng and Pakhi. Nice to see you.
Hello. I won't be joining you next week.
Okay. All right.
Hello.
Hi, how are you?
Okay. Okay.
There you go. Fine. Perfect.
There we go. Okay, good. Okay. So what we're going to do here is very strange. We're going to take a picture and scratch a few lines across it and try to reconstruct the picture just from the lines.
Okay.
Okay. Okay. So the context of this is that we're building up towards algorithms for breast compression mammography, where the X-rays go into the top and hit a detector at the bottom here. Breast will be in between these two plates. And the reason for compressing the breast is that you get a better image if it's thinner. Okay, so usually they compress it until it hurts. The standard seems to be about 5 centimeters. And these things are about 25 by 25 centimeters, so that gives you the size of the volume that we'll eventually have to deal with. Okay? Now, what's happened in the last couple of decades is that a new kind of, excuse me a second.
Right.
I'll look this telephone. Hello? Hi, I'm on the line. Do you need to talk to me right now? I'll call you back later, okay? Bye. Okay. Wasn't that urgent? Okay, now. What happened in the last 20 years or so is that a new source of X-rays has been developed. This is the X-ray generating unit. Can you see my mouse?
Yes.
Yes, okay. The X-rays come in here and they go through a set of glass tubes called capillaries. Okay? Mm-hmm. And then they come out either parallel or focused, depending on the type of tube of glass tubes you're using, or the shape of them. Now this is what it looks like. Here's the X-ray source. The X-rays go into these tubes, and for the case I'm talking about today, they come out parallel. Okay, and what happens is the way this works, the X-rays are very hard to focus or to change their direction. The way this works is these are very tiny capillaries, and the X-rays bounce by total internal reflection inside the tubes. The tubes have nothing in them but air. Okay? This is what they look like if you look at them with a microscope. They're only 10 microns in width, the individual tubes, and there are millions of them. Okay? So you can't see them without a microscope. Okay, so each one is hollow and the X-rays bounce inside. Okay, so what I'm going to do is show you what we've done with a test image. This is what done by Ken Chen and I. Okay, we started with this grayscale picture. Okay, now I have to discuss straight lines across a picture. A straight line, as done naively, is what's called aliased. In other words, if you draw a straight line across a checkerboard, it's a bunch of steps. It can be, in this case, it's 2 or 3 steps. What most people deal with are anti-aliased, where you use an algorithm to make that line look smoother by making some of the pixels next to those grayscale pictures. We're not going to deal with that. We're dealing only with aliased lines. Okay, so we're bucking the trend, so to speak. Okay, so here is that butterfly with a bunch of with 2 to the 5th lines. What's that? 32.
Yeah.
Yeah. Okay, 32 lines, and you can just barely see the butterfly. Okay. Yeah. Now what we did next is we used an infilling algorithm, which is a very simple one. It's let's go back here. If you take a single row across the butterfly with just lines, what it does is each pixel on the line has been extracted from the butterfly. So each pixel could have a different value. You take a row across here, and you take two consecutive lines, and you interpolate between the lines. Just a simple linear interpolation. So you fill in the pixels in between the two lines in the row by interpolating. And this is the result. Okay, so just a simple, and this is a very simple-minded Impainting. Okay, now I wanted to point out that this is the opposite of the general practice of Impainting, at least when it got started. Here's a parrot. Somebody has crudely by hand drawn the cage lines here, and then they inpaint the lines, and you get your parrot without it outside of the cage. Okay. What we're doing is we inpaint between the lines. The lines have the data, and we throw away the rest. Okay. Now, this shows this is a power of 2. We did it backwards here, but with this power of 2, at 2 to the 9th lines, you get the full picture back. But if you use fewer lines, this is the Euclidean distance between the line image and the original. But if you inpaint, the Euclidean distance goes down drastically. Okay, so this gives us a quantitative measure of how well we're doing. Okay, now here, instead of vertical lines, we use random lines top to bottom. Now, the reason we use top to bottom goes back to this slide, and that is the X-rays can only go up and down through this compression plate to the array of detectors, so they can't go sideways. So we're sticking just with the case where they're nearly vertical, but not quite. Okay, so if we go, let's see, if we go here, let's see, there we go. So if we take a bunch of random lines, here again, you can vaguely see the butterfly. Okay. See what happened here. Oh, okay. But if we do our infilling, the butterfly's clearly there. There are obviously artifacts in this infilling because we're only doing horizontal infilling. Okay. And as you can see, we're doing even better here. With the infilling, pulls it down to these values. This is without the infilling in terms of Euclidean distance. Okay. Now, what we're aimed at eventually is what I call ray-by-ray computed tomography. You have to understand that X-rays are ordinarily emitted over a large area. If you've ever had an X-ray, they're sprayed over your body, and everybody gets out of the room. Okay. They may be, that's called a cone beam, where the X-rays come out in a cone. They may be collimated through a lead slit, because lead will stop the X-rays, and then you get what's called a fan beam. And there are thousands of papers on computer tomography algorithms for both cone and fan beams. Rays are what are doing computer tomography with our pencil beams. Okay, so this is a new problem. Now, let's see, next slide. Okay, this illustrates then. Here's a cone beam, goes through a large portion of the patient. Here's a fan beam, just goes through a plane of the patient. And here's our X-rays, which can be aimed in any direction because at least in the initial prototype, we'll carry the X-ray source by a robot. Okay, and it has to be a strong robot. It weighs a couple of kilograms. Okay, so it's an industrial strength robot. Okay, oops. Okay, so what we need is better in-painting algorithms, which it's apparently a very sophisticated field that's gone from differential equation approaches to machine learning nowadays. And we could probably do significantly better than what we've done in like this. So in other words, this curve here with the in-painting might come lower with better in-painting algorithms. Okay, so if anybody's familiar with in-painting, it'd be nice to have. Okay, next question. How do you choose the lines? Okay, and the question is, would image-dependent image processing help? In other words, if you've chosen n lines to choose the n+1 line, do you look at what you've got so far and somehow figure out where to put the next line instead of putting them in random? We tried one way of doing that and it didn't help. So it obviously needs some thinking. Okay, now the other thing is, what's the difference between X-rays and taking a line out of a picture? The difference is very simple, and that is X-rays give you only the average value of all the pixels along the line. They don't give you the individual pixels, okay? And we have to minimize the number of lines because every line is an X-ray, and the more X-rays, the more dose, and breast tissue is very sensitive to problem. Oh, I should have mentioned that 1 out of 8 women experience breast cancer in their lives. Okay, at least according to North American statistics. It may be worse outside of North America. We have to generalize these algorithms to 3 dimensions because we're dealing with 3-dimensional breast. And it's been shown that the normal breast tissue can be characterized by what's called Perlin noise. So we have to see if we can do this in terms of a background of Perlin noise and not a clean background like I've been assuming. Okay, and one problem is the way we like to, if you find a tumor, and we're looking for 2 to 4 millimeter tumors, before this is the size at which most tumors have not yet metastasized, which means they haven't spread around the body. So you've got a chance of curing the patient, okay? And we're thinking of doing it using polycapillary sources, which are focused, and so the glass tubes are bent this way to a focus. So you would aim, the tumor would be here at the focus, you would try to destroy the tumor, and you'd also use the robot to go around and do it in different directions so that you gave as little radiation as possible to the normal tissue. And mostly hit the tumor. Okay, so that's the problem we're aiming at. And here are some suggestions using the MART, probably with in-painting, the multiplicative art algorithm, the power MART, which takes the correction factor to a power, the use of voids, and machine learning. Okay, so we've got a lot to do. So that's it. Okay.
Okay. That's great.
Okay. Any questions? Susan, ouch. Yes. Only breast cancer occurs 1% in males. Because that spreads the ouch.
Yeah.
Okay. So usually they're concerned with females. Males also are very stubborn. They come in late, and it's usually too late to try to cure them.
I know some women that refuse to go get it done.
Oh yes, I believe you. You know, and they're trying to modify those breast paddles so that the force exerted is acceptable.
Well, they have fixed it a bit.
Yeah, but still the images aren't great because they're just projection images in one direction. Okay, we're trying to find tumors in 3 dimensions and cure the patient. The idea is, while you're still in the machine, if a tumor is found, the machine zaps it and you're cured. And so that's the idea.
Yeah.
And there's one paper suggesting that these things will destroy small tumors. So we seem to be okay. I don't understand Pakhi's. What did I say that means that to which you want to reply? Yeah, exactly.
I think
what?
to Susan's ouch.
Oh, yes. Yeah, they're women. Yes, they're getting better at it. But the problem is that if you just take a standard flat X-ray image of anything, or like you're not likely to find. You're not going to find, if you see tumors, you won't know where they are in 3 dimensions. And so we have to go after some sort of 3-dimensional calculation. Oh, Lei Peng can't see you. Okay.
Oh, he's on.
Well, get your camera fixed. Okay. Lee, how do you pronounce your name? Lee or Lai? Okay, not gonna. Maybe it's. I hope you heard what I was talking about. Okay, so that's the general problem. You can see it needs a lot of work. It needs more testing on the pictures, and it certainly needs better in-painting algorithms.
Yeah.
I invented this extremely simple-minded in-painting algorithm because the other ones were too hard for me.
Yeah. So yeah, I had a couple of questions about the slides. So the Euclidean distance is measuring what exactly? Like, what is the distance?
It's just the Euclidean distance between all of the pixels, corresponding pixels, normalized.
Okay.
Okay, we normalized it.
So in painting,
I think the scale is 0 to 1. Yeah, go ahead.
Go ahead.
That back. Yeah, I think it's 0 to 1. In other words, this plateaus. Well, when you have a small number of lines, the average distance is 0.4. That's distant. I think it's the square root of the sum of the squares of the distances between the pixels.
Okay.
Okay.
This, I have had some experience in some similar fittings. If this is the same.
Vahid, your voice is. Vahid, your voice is breaking up. Could you please use the chat?
Yes.
I couldn't understand your voice.
Is it okay now?
Yeah, it's better. Yeah.
Yes, okay. Okay, go ahead.
Okay. I was saying in the past, some of the fitting problems we were dealing with were something like this. I couldn't actually get the right message from what's the problem you are going to solve, but I might be actually helping in an optimization or a fitting problem like this.
Okay, this distance here. Can you still see my picture?
Yes.
Yes. Okay. The distance here is typically a fraction of a millimeter. Okay. So that you're getting a beam of X-rays. Okay. That's what you get in the real case. Now, what we're doing here is not using a beam of X-rays. We're just taking lines extracted from the original picture. So if you extract all of them, you get the original picture, which is this is the original. Okay, if we extract them, then you get these, but you get zeros in between because you haven't extracted those pixels. And then if we do infilling between the lines, this is the kind of picture we get. So you get a tremendous improvement with infilling. Okay. However, there are obvious artifacts here, and you can see the horizontal artifacts everywhere. Okay, and then if
something like it seems something like an interpolation problem, yeah. So here,
this is the power of 2 of the number of lines. We add 2 at the edges so that the computing the algorithm is uniform. In other words, here, if we go back here, there's actually a line here at the left edge and one at the right edge. That's where the 2 to the 5th lines plus the left and right edges. Okay. By doing that, we just have to interpolate the same way here as here. Okay, it simplifies the program. Okay. If we use random lines, this is what we get, as you said. Okay? But still, even with random lines, it's pretty good. Though we had the number of lines went up by a factor of 10 in order to get this. Okay. Now you could use other distance measures than Euclidean. Euclidean was just the simplest.
Yes.
Okay. But it's remarkable how well Impainting does, and then you can see you can see your basically your error here in the average. This is only even at 2 to the 5th here. The average is only 0.1 of a pixel. Okay, so it's pretty good. So the problem is to get this curve as low as possible. I think better Impainting algorithms would do that. This Impainting algorithm, which does it horizontally, is very crude. I invented it myself, and it's obvious. Okay, but even as crude as it is, it's pretty good.
Right.
But as you mentioned, usually the problem is with the right initial value choosing. Yeah. So I have had some actually similar problems that I could solve with some using some IVP better than maybe some other types of interpolation. So if I could delve into the problem, maybe I could help on solving and having a better error measurement. So I would be glad to help me this. This is something that I've been working on.
You want to give me your email address? I can send you the latest draft of the manuscript. Put it in the chat.
I guess you have it, but
I have it. Not in my head. Maybe somewhere in my computer.
Yes.
I was going to ask if you published it yet. Not quite, right?
No, it hasn't been published yet. Worst case, it will go into a book that we will eventually write on this whole problem. But no, it hasn't been published yet. It would be nice to add better Impainting algorithms, see how they work, and if you do that, you'll be a co-author.
Okay. Vahid, there you go.
No, maybe that's a challenge. Anyone could do a better job. Maybe. I'm just challenging myself as well.
Okay, that's fine. Okay, so the basic difference here, then, if I go back to the line image, the basic difference is that along each line, instead, you see, if you look at an individual line here, you can see the density varies. Okay, with X-rays, you only get the average of all the pixels along the line. That's the difference, and that's where we have to get into algorithms. And maybe Lee will do that. He has experience in computer tomography. Okay, so that will be a separate paper. That's why I call this paper number 1. Okay. Okay.
And, sorry, okay. Just wanted to mention that I have prepared something as well. If there is any time at the end, I would be happy to present.
Oh yeah, that would be great.
Yeah, I've been talking to Bradly in the past.
Yeah.
Yeah. If there's any time,
Oh.
if not, I could postpone it to the next week. Yeah.
Okay.
When you get it to the point where it's published or something, maybe you could present this to the biomedical engineering seminar.
Oh, sure, but it's hard to fill an hour with. Well, yeah.
No, it's 3/4 of an hour, and then.
Yeah, we have pictures for all of the numbers of lines that we used. Okay, so I've got about 100 pictures. I really cut them down for this presentation.
Yeah, well, I mean, when you worked on it further and have some 10 more slides or something, it probably would.
Well, I could do that. Yeah, could do that.
Oh, I just thought that it would intrigue the students.
I was trying to keep it short and sweet here. Okay, just single examples.
Well, this is simpler than what often gets presented to us.
Oh.
It's more understandable, and I've been in lectures that I understood because I understand some biochemistry and a lot of biology.
Yeah.
And the rest of the students who are mechanical engineers are going, what is this?
What is right? Okay. Well,
understanding
this is assuming some image processing, I guess, so has some experience. Like the difference between an alias and an unaliased line.
Yeah, I do know that.
The reason we use the alias line is because it allows us to just sample the picture easily. If you used an anti-alias line, you'd sample them with weights. It would be loose. Okay, so we did the simplest case. Okay.
So I wonder if you could make a few words about the Perlin noise simulation. So we had discussed that a while back.
I've got a paper which claims there are 3 kinds of tissues in breast, and each one has its own Perlin noise with different parameters, and I haven't been able to figure it out yet. Okay, if you want to delve into Perlin noise, you have to understand this paper and see if you can figure it out.
Yeah. So why does it have? Is that like a background noise for the image?
Yeah.
I mean,
well, they claim that the noise looks the same as normal tissue.
Oh, okay.
Okay, so what we would do is then start with a picture with Perlin noise in it. We would add a simulated tumor, and there are lots of papers on that, on how to simulate a tumor. You can do it simply as a disc, a filled-in circle, or they've got more sophisticated ones where they look at the properties of tumors. And then the question is, can you detect the tumor this way using that Perlin noise background? And assuming that that these people are correct, that they have characterized normal breast tissue as looking like Perlin noise.
Right.
Okay. Now, if you're interested, I can send you papers on Perlin noise.
Yeah.
And this paper that claims the breast looks like Perlin noise.
Yeah.
Okay. They're more sophisticated. Yeah, I only put down the simplest problems here at the end. For example, are any of you familiar with K-edges? You're all shocked.
No, not necessarily. Yeah.
Okay, a K-edge is typically done with iodine. Iodine apparently concentrates in tumors. And this is just, I think it's just elemental iodine. And it absorbs X-rays, but it has a remarkable property that at a certain wavelength, the absorption of the X-rays suddenly jumps. So if you use one wavelength of X-ray and then the other side, that's called the K-edge, and then you get 2 pictures, and then you can look at the difference picture between them. So we may have to do this with difference pictures to get rid of the Perlin noise, for example. Okay, so that's called K-edge imaging or differential imaging or whatever. But you can locate the iodine gets into the tumors, and then hopefully that would help find the tumors. Okay, and one of the problems we have is that the physical properties of these tiny tumors, these are 2 to 4 millimeters, premetastasis means before the tumor spread to the body. The physical properties of these are not well known. Okay, so we're guessing at them. Okay, so that's basically there are a whole bunch of additional steps which require a lot of programming, and we're trying to do the whole thing by simulation to see if it works. And we're only simulating physical things, so it should be possible. Okay.
Yeah.
Okay, everybody know what voids are?
Well, we've talked about them in the group, but you might want to.
Yeah. Voids are simulations of flocks of birds or schooling fish or something like that. Okay, they can be used. The idea here is to move the pixels around as if they were voids. Voids represent birds, computerized birds, but we'd have to move them back and forth on the lines and let them fly on the lines and move the densities around. Remember that with the X-rays we start, every pixel initially has just the average value. But as the lines cross, you can improve the value. So one approach is to use voids to fly them up and down the lines until we get a consistent set of voids representing the densities along each line, and then do interpolation. And in machine learning, on this problem, I don't understand how it goes. I mean, somebody who is into machine learning needs to look into that. Okay, so it's sort of an open-ended problem. We think we can solve it, and these polycapillary sources now make it physically possible, and they're easy to simulate. So we think it might be possible to do this problem. Okay, so we're after the aim of the ultimate aim here is to search out tumors, find them, and destroy them, and hopefully cure the patient. Okay, so Susan, for all the pain, if this works, you go into the machine, and if you find something, you come out cured.
Yeah. Okay. I still think there ought to be a different mechanical way of doing it. I keep saying, well, it was designed by a mechanical engineer. What do you expect?
Yeah, you give a woman a microphone, and when she says, ouch, you stop.
Yeah. Like I said, they have improved it a bit. So.
I mean, that's basically what it's about.
Yeah.
But the standards squeezing of the breast seems to be 3 to 5 centimeters.
Yeah, well.
and it does have a reason. And the reason is that you, by going through less tissue, the X-rays give you better images.
Right.
Okay. So there is a rationale to it. Now, there are some sadistic techs who will squeeze to make sure it hurts.
Well, no, like I said, the last time I talked to a tech, she said that they had improved it a lot. So that's good.
Okay, yeah, it's coming along, but the pictures aren't getting any better.
Oh, right.
The problem is very simple. You see, computer tomography we're all used to now. If you need three-dimensional imaging of part of your body, you go into this $1 million machine and it'll make a nice picture for you. But the methods that are used for breast imaging haven't caught up with this yet. The most they have is what's called tomosynthesis, which is a very crude form of computer tomography. Okay, so what we're after is doing it right and doing it effectively. Okay, and the reason for this is, if I may make it might seem like a sexist remark. I've been involved with groups of women who try to raise money for breast cancer research. And what's obviously going on is that they raise the money, they give it to the men say thank you, and then go away, and they will not talk to these women again. So the problem we have is there are very few women who seriously get into breast cancer research. And I've seen that problem over the decades. This problem is not going to be solved probably until women get involved.
Okay, well, there's becoming more and more women biomedical engineers. Like,
Fine. I haven't seen any in breast cancer yet.
okay, well, they're,
Well, not quite. There's a woman named Carolyn McDonald who's shown that she wrote one paper which shows that the focusing at polycapillary sources have sufficient energy to kill tumors.
oh, okay.
Okay, and then she did nothing further.
Oh, well, I have had that done with some of my research.
Yes.
I just wrote one paper. It's quoted over 150 times, but I didn't get to do another one.
To get somebody to follow up and do the hard work and say, okay, so let's actually kill some tumors.
Yeah.
But,
yeah.
She didn't go for 3 dimensions, and I've tried to get her involved. She won't. She's not interested. So, anyway, but the focus. So, but at least she's shown that the focusing polycapillary sources are sufficient to kill tumors. The energy of these things has come up.
Oh, well, this would be a reason for you to present to the biomedical engineering students.
Sure. Just suggest a date.
Okay, well, I have to persuade a certain Dr. Sharif that this would be interesting.
Oh, okay.
Yes.
Oh. Sharif is here. Is at the University of Manitoba partly because I visited him after his recruiting talk and tried to convince him to come. So.
Oh, okay. Well, I remember in his introductory speech, he defended my master's thesis, and he didn't know he had. It was good.
Okay. Anyway, if anybody wants to help, we're doing the whole thing by computer simulation, as I mentioned, including the robotics and whatnot, because we can't afford anything else. These new polycapillary sources are cheap compared to mammography units. Mammography units go for $125,000 to $250,000. These polycapillary sources are about $30,000.
You didn't mention that in your talk to the VME. I'm gonna.
Oh, okay. Cool. The relative cost. So the polycapillary sources are practical in the concept of what kind of money is being spent now. Okay, and if this works, it could help 500 million women in the world.
How much are they again?
30,000 for the X-ray story.
Oh, okay. Polycapillary are 30,000.
Yeah.
Okay. Price of a car, sort of.
I think one of the reasons they're so expensive, it's really finicky to bend that glass so it's got the right shape.
And how much are the other ones again?
Well, a standard mammography unit, as used today, costs up to $275,000.
Oh boy.
Okay, so you see, we're we're well within the ballpark. If this works, it's buildable at a cost probably less than that of a standard unit. Okay, now there are all sorts of problems. Right now we're carrying the X-ray source with a robot, which means the speed of the robot and how fast it settles down when you move it from one position to another is probably going to be limiting. Anybody with robotics experience maybe could help out and make some realistic estimates. I think the whole X-ray steering eventually, the aiming of X-rays in a parallel beam in any direction you want can eventually be done electronically, but that's going to take some new technology. Okay, so I think I regard the robot carrying this X-ray source as a prototype, not as the ultimate. It's a proof of principle, so to speak.
Oh, okay.
Okay, because I think it's going to be slow, because a robot that can carry a couple of kilograms is not going to be a fast one as far as I know. But okay, you know, most home robots can only carry a few hundred grams, not kilograms. Okay, and obviously this programs the robot, making sure the robot doesn't damage the other equipment, and make sure he doesn't damage the patient. Okay, so there's some clever things that have to be done with the robot. Okay, any more questions?
I think that's it. Yeah, so thank you, Dick, once again for presenting on that.
Okay.
Right. Yeah, good. And then Vahid wants to present something. So would you like to go ahead?
Okay.
Hello again, everyone. Yeah?
Yeah.
Actually, this is something we were discussing in the past with Bradley. Let me share my window.
All right. All right, there we go.
See my, yeah, okay.
Yes.
There were some questions I had in mind that based on them I was going to have maybe a review paper or a new kind of work. I've been talking to Bradley during the past week. I've somehow managed to try to have some questions and some available works, and so what we could do by using those kind of questions and works. The main question actually is based on a developmental perspective to artificial neural network models that basically could be derived from C. elegans, but also we could have some type of work that could help to better understand and better simulate what type of actually developmental processes could be actually better simulated in C. elegans models, especially in the locomotion of C. elegans, as we have done some great works in the past in the OpenWorm project. So the main questions, I mean, maybe they have been answered in the past in the OpenWorm project, but I'm trying to look at them from different aspects. The first question is that what could be actually the main milestones for C. elegans to become an adult from a locomotion point of view? If there are, for example, different days that could be considered as the main ones to be actually distinguished. We consider distinguishable movement patterns, and so if that's it. So what change could be recognized in the neural networks, and so what change could be applied to the equivalent electrical circuits that so far we have simulated. Similarly, what could be recognized in structural compartments, those that could be again applied to mechanical models of the movements, and so if it is it possible to derive some new artificial neural network based on these developmental chains? And on the other hand, could we have a chronological history of development of artificial neural networks that could be helpful? A history that I'm going to mention somehow using the references that I've exerted so far, and also from a thermodynamics and energetic point of view, what could be considered as the main state, and what other environmental factors could be affect the model. These are maybe the main three categories that could be considered as the different maybe milestone for new work that or a review work that could be done. I'm just saying that it's a new work I've started. Maybe it's for 3 or 4 days that I've been delving into this. So I would be glad if you know any works that have been done, specifically in the Open World project. I've read and somehow exerted some of those works over here, but I would be glad to hear your opinion as well. So, based on these questions, what we have currently in the Open World project is a movement simulation that has been simulated through the cybernetic project. That has been simulated by mechanical circuits and connectomes. And also the current mathematical models and simulation for ionic currents, especially muscle bodies, and for an adult hermaphrodite elegans that is based on the work by Boyle and Cohen in 2008. This is part of the project that has been simulated, and we have the codes both in MATLAB and Python at the Open World project. This is a model that they have mainly the work is based on. There are some different simplified models of arms and also some circuits that have been equivalently designed. There are, I guess, 29 parameters in this model that they try to simulate the model, and this is the simulated model of a current that we could refer this as well over here. So the main thing, the starting point of this work is how we could have actually different stages of development in C. elegans locomotion that could generate some similar patterns and models like these for different maybe states. For example, we can say the first day of its movement could be the first key points, and after that, every different pattern that could be recognized would be a new model, and so a new set of parameters. So what we need is some data for structural development and movement of C. elegans during these different stages. And so after that, fitting some new parameters to those, actually to the above-mentioned model, to find out new models for each stage. These are the main maybe points that I'm going to refer during this work or this review paper. I started with some reviews for what have been done in the past, some from maybe early days of the works with Asparis worm nematode, and also after that some breakthrough in the C. elegans. I tried to find some main papers in structural developmental and especially in modeling references that we could have, and also some other related references, adaptances, and some foreign computation works and what we have done in the Open World so far. These are going to be the main references that maybe we could collect some of excerpts of them, and I try to have some chats with ChatGPT as well, and asking for some questions. So I try to have some cheap applications. I couldn't. I don't know why I couldn't find any related work to the Open World project during all my questions in ChatGPT, but we might get your work in the future why this is happening, and nowhere is mentioning actually the Open World project as a source of some computational models of C. elegans. So I started with a chronological review of nervous system development in C. elegans. I could have some first initiatives, and after that, one by one, actually making it improve. And finally, I can say I could have this main work, some functionalities that directly from series that I'm going to improve the model, and also one which is related to what I've been talking so far, some differences, and yeah, that's it. That was the key one. Yeah. So for the first part, I was interested in knowing if anyone at the DevoWorm project, because I mainly know that at the DevoWorm project we were more focusing on the embryogenesis from a actually a genetic point of view. That's when an egg hatches. After that, what would happen? That is somehow maybe the worm grows up and become an adult. But what happens in between? I mean, from a behavioral point of view, for example, the behavior like a locomotion, what happens? Actually, I was wondering that is there any computational and mathematical model for those kind of developments that could be considered as a maybe any work or something that have been done in the past for such computational models?
Thank you. Yeah, well, that's a very complete document. If you could send me a link to that, I'd like to take a look at it.
Of course.
As we discussed. So yeah, it's great. There's a lot of stuff in there I see like that are potential things to focus on. And then the thing you highlighted last, which was like what kinds of things are happening from like the egg, fertilization of the zygote to like, the hatch of the egg or even to the adult. We kind of know what those things are, but I don't think there are any mathematical models for that. So, one thing is like to look at like ion channels as they change from like a undifferentiated cell to like a mature neuron. And there's there's an area of work on bioelectricity that people have done with not just with neurons, but with other types of cells. So all cells, even stem cells, will express ion channels of different types. And those are usually used to keep, to regulate the electrical activity or the physiology of the cell. Now, in neurons, you get very specific neuron channels that are of type that create an action potential. And in C. elegans, we don't necessarily think it's a full action potential, but the point is that you get this expression of ion channels through development. And so I don't think people have characterized what those are. Like the progression from, say, like any one stem cell or precursor cell type to the adult form of a neuron. But, that's something, that would be interesting to simulate because we have in OpenWorm the tools to simulate different, neuron channel or different ion channels and how they affect the cell and what kinds of potentials they generate. So that might be an interesting thing to say, like, well, we can make some hypotheses about what gets expressed in the cell. We know maybe what stem cells are expressing. We know what maybe adult cells are expressing, and we can work from there. So there's an ion channel component to that. There's also maybe like a behavioral component, which is that behaviors, start with like these sort of endogenous movements of the embryo. So like there are movements in the embryo, like you get a comma stage of the embryo where, the embryo starts to take on a shape. You can see it in the microscopy of it. And then you have these other endogenous movements like muscle twitching. So when you get the formation, you get cell migration, early, and then you start to get like the formation of muscles. You get muscle twitching, but it's not like controlled by the nervous system. It's controlled just by like its own electrical activity. And then you start to get the brain or the nervous system wired to the muscles, the connections make it to the muscles, and then you start to get maybe some behavior. And so, it's kind of interesting that progression of different movements from the embryo to the actually we end up with the first larval period. So in L1 or the first larval period is where we start to get these sort of movements that are autonomous and actually are directed towards like food or something like that. But yeah.
But mainly, if I'm going to point to one main question is that do we have any type of data that has been somehow recorded the movement of the C. elegans in different states, and so and also recorded the electrophysiological maybe characteristics of those movements, something that has been done in this work by actually the data is another work, but the model has been produced based on the data that says how that actually shows how the recorded movement of a C. elegans could be matched to the electrophysiological data like this one.
Yeah.
But this is for an adult. Yeah, this is for an adult C. elegans. Do we have any similar data for other actually developmental? Have you ever seen? Because I couldn't find these 1 or 2 days I've been searching for. But have you ever seen any similar data for other actually developmental stage? For example, C. elegans in day 5, in day 10, in day 15, and lastly in day 20. For example, if we could consider 4 developmental stages and different movement and maybe different ones. But is there any similar? Have you ever seen any similar work that have been recorded those movements in different states? And also, they beside that, the more important one is the electrophysiological characterization of those action potentials, movements, and especially ion currents.
Yeah. No, I know that they're like, I'm not sure about like the larval stages. So, the thing that I guess you'd be looking for would be like people recording neurons in larval worms from L1 to L4 and then like looking for differences. Yeah. Now, I haven't seen anything like that. It may exist, I don't know. But one of the interesting things is like there's this stage called the Dower stage, which starts at L2 and it goes to L4. So it's like this alternative sort of diapausal state. And in that stage, you get a lot of like, sort of this quiescent behavior. So everything is slowed down, the metabolism slowed down. And I would look and I'll look for this. There may be people who've done this in Dower stage where they can look at like the neuronal activity. And I bet you that would probably show some pretty interesting differences. But across like from L1 to L4, I'm not really sure. And it may show some differences, but I don't know if people have done that. So.
Okay. So going back to the main question, I was asking this question because I was more interested in the artificial neural networks derived from these actually models. So if we could have some, it is something not natural, but maybe natural developmental process that the learning of something new is going to be happening, something that has been coded in the C. elegans and it's going to be revealed after some developmental processes. So we are going by this one. We are going to mainly understand what new type of artificial neural networks could be derived from these developmental stages, maybe stage by stage, and then by fitting those new parameters to a model like this, we could understand maybe a new type of learning system that is actually is happening right now in the elegans from a locomotion point of view,
Right.
and that could be again extended to other type of behaviors.
Yeah. Well, I mean, I know that people probably haven't done that in terms of, I know people are starting to go in the direction of like building artificial neural networks that conform to certain, like they talk about how it's anatomically sort of grounded, and they'll do things like have different layers representing different parts of the brain anatomy or something. It's very general in terms of the metaphor. But I've not seen anyone do anything developmental like that, generally speaking. Now, you bring up a good question, which is, to be able to, say, take a artificial neural network and maybe like evolve it in stages or have different stages of its development. So what that means is that you would have like neurons in the neural network that would maybe like be born and have certain properties, and then over time those properties would change and you'd end up with a mature state of the neuron, and that would just that would affect the information processing, and there'd be a lot of interactions there, which would be we could play around with some toy models maybe to start, which would be like very small neural networks, and then figure out like if we have neurons, and this would be maybe after we look at the data and see we'd have to kind of define the stages first. And then you'd have your artificial neural, it'd have a toy model, which would be a very small network. And then you'd say, okay, now how do we want to implement these neurons? How would they, might they be able to do some standard computational operations, like logic gates or something like that? And if they can do that, then what is maybe the advantage of having those developmental stages in there? What would be if you switch on like a neuron or change its developmental state? What is it doing in there, and why is it doing it, and what's the advantage of that? You can just simply model development, recapitulate development in the neural network, but with a neural network, people tend to ask like, well, why do I want to do this? So it's kind of pointless otherwise.
Yeah, that's an old question. We are not going to actually to maybe derive a new artificial neural network that could be could work better than what we have right now, because for sure what have been done so far in artificial neural networks is based on some much more complex creatures, like maybe humankind and their neural network that is billions of times more complex than C. elegans. But the main question that we are going to maybe in such a paper we're going to ask is to understand how that maybe learning process in a C. elegans is actually happening, and some questions like that. But from as for me and maybe for others, much more from I'm trying to based on another work, I'm trying to design some mathematical models for these developmental processes, so that it could show how a simple model of maybe a circuit with just maybe 5 or 6. Here we have in our circuit we have 3 compartments, maybe from some simpler one to a more complex one. In a C. elegans, it could really greatly show how a mathematical model and an engineering point of view could be derived from such a simple model.
Yeah, I think that's all interesting stuff. I'll have to send you another paper that we've been working on. This is in a different group, where we've been looking at, there are these, like, I guess they're called like epigenetic functions or something. I'll have to dig it out, but like basically where you turn things on at different stages. It's like a computational function that, like, this is something that people have explored in the artificial life literature, but it basically is a developmental, it sets a developmental set of stages for artificial neurons and other things like that. So I'll have to, it's related to that, Jesse. So yeah, I'll send you that paper and I'll send you more detail. I can't remember the exact details right now, but yeah, that's an interesting area. I think there's a lot of work that could be done in it. So let's keep thinking about it. And please send me a copy or a link to this document.
For sure. Actually, I was going to a little more purify the document and then share it with anyone who's interested. I will do this in a day or two as well.
Okay. Yeah, it's great.
Or maybe today.
Yeah.
I need a little exertion from everything I have collected so far.
Yeah. All right. That sounds great. We did have some other things in the chat, like some side conversations in the chat about Dick's talk and then some of the other things. So we had a question by Pakhi who had to leave. So I had a question. My network is unstable. For the breast cancer problem, are we using a machine deep learning to get a better version of Impainting algorithm? Yeah, so that's what Dick was saying, is that we don't have a good machine learning solution, and machine learning could help, deep learning could help, but you'd need, of course, training data, and then you need to have, basically what we would be trying to do there is find features in noise, or extrapolate features from like a very sparse data set. So that would be the way that you would do that, and then you'd have to apply the algorithm. Susan says a better version of Impainting needs to be developed. Yeah, we talked about that. Perhaps a machine learning version would work better. Yeah, I mean, for that, I was looking at like how they were generating the lines, and there were these random sort of line strategies, and there are different ways you could generate the distributional lines. I mean, there are different distributions you could use. It kind of reminds me of pickup sticks. I don't know if you ever played that when you were a kid, where you have a bunch of sticks, you throw them down, and they form these patterns, and you pull them apart. I mean, they form different patterns, and they're kind of random, but you can generate different like patterns of lines. You can generate generate from one side then another side, and do it that way. And that might give a little bit more information. But yeah, there are a lot of strategies one can use algorithmically to do that. And then Dick says, Pakhi, responding to Pakhi, there are CT algorithms that use machine learning and others that are for Impainting. My talk today was on precursor, reconstructing a picture from lines of a pixel extracted from it. So yeah, that was what we were talking about. And then, so there was another, this was a Zoom link. And then Jesse said, this is a different conversation. This is the second conversation with Vahid. The topic of stage differentiation is interesting. That's great. And then, this is, thanks for all those comments. So let's follow up on that stuff. I know Dick has been working on this for quite a while, so it would be interesting to see people kind of take on some of these projects. And, I know that Dick wants to publish some of this work in different guises, under different guises. So that's an opportunity to get published.
Artificial life, is that another method of modeling cells and their interactions?
Well, it's a field where people basically tried to simulate life. So, in the field, they do a lot of things with simulating cells, simulating genomes, simulating evolution. A lot of, even some people doing things outside of like computers in terms of like working with protocells and things like that. There's the A-Life conference link. So that, they have a lot of different things, but there's been this sort of imperative to sort of simulate life. And people have focused on, largely on either like, biologically inspired models or like genetic algorithms or things like that. And from that, you get these innovations. Not a lot of people have done things with development. So there have been different, people have been trying to develop different tools to work on development. But yeah. Oh, I think the term I was thinking of was a canalization function. So this is from Waddington's work on canalization of development. So they have these canalization functions, which are where you kind of take a certain trajectory, and then halfway through the trajectory, you switch to a different trajectory, and you keep switching down until you get to an adult form. So the idea would be that you would have one set of like rules for your artificial neuron. You'd switch to a different set of rules, and then you keep doing that until you get to the adult form. And the idea would be to recapitulate what's happening in development, where you have different cell types that change. You go from a precursor cell to another type of cell to an adult form. So there are different things happening in development that correspond to sort of the development of that cell. And you could just say like that could be something like a set of logic functions or a set of ion channels that's expressed to give a certain output. So that's what that would be. And so it's a computer. It's in a computer. We're not trying to take every part of development and model it. We're just trying to model the most important parts for getting a sort of a computational output. So. Okay. Well, thank you for attending the meeting, everyone. It was very interesting. A lot of good things going on. Are any other comments or questions before we go?
No, I'm asking about the artificial life because I wonder if I should include it in methods of modeling cells.
Oh,
but people have used it for that. So
yeah, there might be some good references. If I don't know. There are a lot of conference proceedings. There's an artificial life journal, so there might be some interesting articles there.
Well, I'm just going to put a one-liner in there saying This is another type of model. Yeah. And if they want me to expand on any of my sentences in my 40-page write-up, I can do that.
Yeah. All right.
Anyway, yeah.
Yeah. Okay.
Also, I've been talking about some part of maybe some point that is more related to the thermodynamics and energetic point of view of these developmental processes. So maybe if Susan has some point on that, I would be glad to share with her. I've been trying to contact you in the past because you've been talking about some biophysics, biophysical maybe perspective of the works, but couldn't find your.
Okay, yeah. On my email.
Yeah.
No? Yeah. Okay. Sure. That works. Do email.
Glad to be in contact.
Yeah.
Yeah, for sure. And of course, Dr. Gordon, if you could help, because I know you have been doing some great work in the past. I've read some of your work. I would be glad to collaborate with you as well.
Okay. Just write me and let me know what you want.
Yeah.
For sure. Thank you.
Okay.
Thank you.
Thanks, everyone.
All right.
All
right. So now I'd like to talk a little bit about different developmental stages and simulating things in an artificial neural network, and then some of the canalization functions and what those look like. So, we talked about development. We talked about having, say, like a precursor cell. And C. elegans, which you'll typically have as what they call a developmental cell. And that developmental cell will have a nomenclature of the lineage tree, so that could be like ABAL, for example. These cells will they keep dividing and they form adult cells down the line. It could be adult neurons, it could be adult epithelial cells, they could be a number of things. And so we know what the fate of any one of these precursor cells are in C. elegans before they become an adult cell. So you have these different cell types. Epithelial, muscle, and their different lineages. Actually, muscle does not come from the AB or from the AB lineage. But anyways, you have a number of different cell types that you can have coming out of a number of precursor cells. So one of the things we were talking about is ion channel expression. So in these precursor cells, you have some ion channel expression and you have some electrical activity, although it's not as defined as in the adult cells. So like in neurons, you have specific ion channels that are expressed for different types of neurons. In epithelial cells and muscle cells, you also have other ion channels that are expressed. They're very distinct to the cell type. But in the precursor cells, we can't say that those ion channels are being expressed. So you might have a subset of those ion channels expressed. You might have different ion channels expressed. I don't think anyone has characterized this in a data set, but we can do simulations where we presuppose that the precursor cells will have different ion channels, like maybe a subset of ion channels, and then more ion channels get expressed as the lineage develops and you get terminally differentiated cells. So, this is a set of experiments that we could do because there's a whole literature on bioelectricity, sort of what happens if you have developmental cells that have this subset of ion channels, what do the electrical signals look like, and maybe what happens when different neurons are connected. And so what happens there is you get these developmental cells that start to differentiate into terminally differentiated cells. And in doing so, they start to form a neural network, so they start to connect via axonal connections, or sometimes there are gap junctions that make shorter electrical connections between the cells. And so in the case of our ion channels, those drive ion channel expression. So when a terminally differentiated neuron synapses with another terminally differentiated neuron, or they make these electrical connections, that triggers ion channel expression that then can change the function of the cell more completely. So you get this, I guess, electrophysiological phenotype that defines developmental cells, that defines these terminally differentiated neurons, and then defines the connected once the neuron's fully connected. So there are these different stages of sort of maturity to this emerging neural network. We have sort of this first phase, which is the developmental phase, we could call it, or the embryonic phase. And this is where you have your lineage tree. And those lineage trees, or those developmental cells in the lineage tree, will express like a subset of neurons, or a subset of ion channels, or they could express different ion channels altogether, because developmental cells, like stem cells in mammals, have ion channel. They don't make action potentials, but they have electrical activity that affects the physiology of the cell and maybe neighboring cells. So we have this developmental embryonic stage. Then we might have this sort of maybe mature stage where the cells are mature, but the network is not. So then we have this third stage, which is the mature network. And that's where you get this modification of ion channel expression not through the maturation of the cells, but maturation of the network connections. So those are our three stages. And so, we can model it that way, and we can electrophysiological properties for each of these. Okay, now let's talk about canalization functions. So this is something that we've explored in the context of embodied agents in some of the work we've done on developmental neurosimulation. So these are canalization functions. So canalization functions are based on Waddington's work on canalization, cell canalization and development or canalization networks. So what I'm going to do is I'm going to draw an epigenetic landscape. And this is from Waddington's work. So, this is something that people have developed quantitative models for. Waddington was really thinking more metaphorically. So you start at the top of the landscape with, usually uses a ball to show the trajectory. And there are these, what they call channels or canals. And so canalization is where the ball will follow a certain path. through this landscape. So oftentimes you'll have these branching points that define different paths through the landscape. So you'll have like these different paths to a mature phenotype. And again, this is a conceptual view, although people have put numbers on this and quantitative values on this. And so this is what the landscape looks like. It looks like a tree. And it's just these channels through this landscape. So it cuts through the landscape to finds a minimum or a minima path through the landscape. So this ball will roll down this path until it reaches this branch. And this branch point is important because it acts to switch the ball from one canal to another. So depending on if you have two different species or if you have two embryos under certain environmental conditions, you can either go down one canal or another canal. And so this is, you might think of this as like the path of least resistance. But the important part is that there's a switch at this branching point that will determine which path this ball goes down. But notice when you go down one path versus another, you go to a different part of the landscape. So if we think of this bottom layer here as the phenotypic space, you're in one part of the phenotypic space versus another. And this is a very general model, so it could be a trait-wise sort of landscape, or it could be a whole embryo sort of landscape. But the point is that you take different trajectories given a single decision point. And so, if the decision point is farther up in the tree, then it results in a more radical shift in the phenotype. If it's down further here, like if you have a decision point here and you move this way and you keep moving this way until you get here, and then there's a shift down this way, this results in a lesser change in the phenotype. So for example, let's say we have an embryo, and for that species, the canalization function tells the embryo to sort of take a left, take another left, and then take a right instead of a left. And so, the wild type might be here. So it requires 3 branching points and sort of gives a direction along that landscape. But then there's some mutant phenotype, which is here, and that might be, the instructions might be take a left, and take another left. So the mutant phenotype is here and the wild type is here, and you can see there's a difference in there in terms of where they occupy phenotype space through this single switch that pushes the phenotype away from that wild type to this mutant state, but it follows this alternate pathway, this alternate canal. So canalization functions are based on this logic. It's basically a function that allows sort of an artificial genotype to be regulated in such a way so that it changes the trajectory of gene expression or of the expression of the trait to go down an alternate path. We can think about the phenotype as an artificial chromosome, where the properties of the phenotype are encoded in this encoding here. So this is a gene, and it's composed of zeros and ones, which encode a trait. So say I wanted to have a striping pattern. I wanted to generate a striping pattern from this binary string. I might have a volume here. And then these zeros and ones represent parts where I have sort of a clear surface and a shaded surface. So the ones are shaded, the zeros are clear. So I have a 0 compartment, a 1 compartment, a 0 compartment, a 0 compartment, another 1 compartment. So I have a shaded area here, 0, 1, 0. So this actually gives me a stripe pattern with 3 stripes. And so I can actually form a pattern from this information in the gene. But now see, I want to change the sort of the patterning structure. So I'll have regulatory elements upstream of this encoding. And this genotype, artificial genotype, is encoding the phenotype. So this string is encoding a phenotype. So this string is encoding a phenotype here. Now these regulatory elements can serve as the switches. So this might be switch, we'll give the switch different states. So we'll say that this switch is 0, this switch is 1, this switch is 0. So each of these states can act to change the point at which we, or maybe the states at which we're interpreting this striping pattern. So say, for example, if this regulatory switch were 0, then it would give me this. If this regulatory switch were 1, it would invert these values so that I would have a very different phenotype. In fact, I would just be able to switch the shaded areas with the clear areas in this example to get the output of a 1 in that position. The second regulatory state is a 1, and so that might be like if this were 0, it would write this out, and if this were 1, it might not write this out. So we would have no information and we would have no striping. The third regulatory element might take the values and transpose them, so you might end up with these 4 elements being read last after these elements. So you can see there are a number of things you can do with these regulatory elements vis-a-vis this information in the gene or in the artificial genotype. If we think about this in terms of a canalization function, this translates into switches. So we might have like a switching situation where we could use all 3 of these regulatory elements in tandem and come up with an answer. So let's say, for example, we had our 0 and 1. So our 0 and 1 means that, we can either write, well, I'm trying to remember. So we can use these in tandem. So we can say that if it's a state of 0, it goes in this direction. If it's a state of 1, it goes in this direction. Then if we have a 0 here, a 1 here, a 0 here, a 1 here, and we can just keep going down like this. But the point is that this tree represents different sort of states of this regulatory system at different intervals. So we'd start with the left. We'd say this is a 0 or 1, this is a 0 or 1, this is a 0 or 1. So let's say we had a regulatory state of 0, 1, 0. We would go 0, 1, and then 0. And that would be our part of our phenotypic space that we'd inhabit. And as I said, we have the consequences of this structure would be that a 0 would mean that we would write this information out. A 1 would be that this is the right patterning. So it's not inverted, it's this pattern. And then a 0 means that it's writing it in this order. And so this is the part of the sort of the phenotypic space of this landscape where this pattern would inhabit. If we changed our regulatory elements, we could transpose this pattern, end up with an alternate phenotype where we're reading 1 0 1 0 0 1 0 0, which would change this patterning structure. Or we could have a state where one of these regulatory elements would switch their state, and we would end up with no pattern at all. And so all those things occupy this phenotypic space. And so this canalization function is just basically a set of switches that enable these binary states in this genotypic encoding to be read in different ways and produce different forms of patterning. So it's a form of sort of digital morphogenesis that isn't, based on Turing morphogenesis or anything like that, but instead is based on these binary decisions that get made in the switch. And so this is a computational structure that allows you to explore different parts of the phenotypic space. Another way to think about this too is by using, instead of 0s and 1s, using logic functions in these regulatory elements. So you might have an and, an or a nor, and that might translate into like doing things with this genotypic encoding that, you have 2 genotypic encodings, so you have an alternate encoding where you do like mathematical operations on, bitwise mathematical operations. So you might take like this one where you say, what's the state of both of these? If they're both 0, then it's a 0 output, otherwise it's a 1 output. You might say like, a NOR operation conducted on this set of binary states, XOR operating on these set of binary states, and so forth. And you can define these all up in this regulatory region, which give the instructions for interpreting this genotypic encoding and turning it into a phenotypic encoding. And then we can translate that into a decision tree, which constitutes our canalization function. And so we've explored canalization functions, like I said, in some of the work we've done on developmental neural simulation in the orthogonal lab. And so please check those out for more information.